Review of Chapter 7. Chapter 7 is on cell membrane structure and function. Page 128, actually 127 and 126, show you structures of the pictures of cell membrane structure. The cell membrane itself surrounds the cell. It's about 8 nanometers thick. It's selectively permeable. Selective permeability means that some things can go through, other things can't go through. Think of a chain link fence. Golf balls can go through, basketballs not so easily, selectively permeable. The fluid mosaic model is a description of the cell membrane structure. It's only been a model in the science community since about 1970. The structure of the cell membrane includes a bilayer of phospholipids. Now remember, you know that phospholipids form a bilayer when in water because of hydrophobic hydrophilic interactions. But the membrane also contains other molecules like proteins, glycoproteins, glycolipids, cholesterol, and things like that. Now mosaic means mixture. Fluid is exactly that. These molecules in the cell membrane aren't bound to each other. Okay? They're free to flow, but only within the confines of the membrane. Fluid mosaic model. Now, Roman numeral 2 in our notes talks about selective permeability. It depends on two things: the permeability of the phospholipid bilayer and the function of the proteins in them. Now, things that can diffuse through the phospholipid bilayer include other nonpolar molecules. Hydrophobic molecules can go through. Small polar molecules like water, dissolved gases like oxygen and carbon dioxide can go through. Things that don't go through the phospholipid bilayer very well are larger polar molecules and ions like electrolytes, sodium, and so forth. Now, the transport proteins are proteins in the cell membrane that allow things to go through that otherwise couldn't go through very well. And if these proteins can open and close, they can control what goes in and out. So really, when it comes to the function of a cell membrane, the main function is cell transport, meaning governing what goes in and what goes out. That's what cell transport means. Now, cell transport can be passive or active. Passive transport means it happens on its own. The cell doesn't have to make it happen. The cell doesn't have to use any ATP energy to cause it to happen. Active transport means the cell has to make it happen. It has to do work. It has to use energy. It has to use ATP. Now, Roman numeral 3 discusses passive transport. I list several things which are all really basically the same thing, looking at it in different ways. Diffusion, osmosis, and facilitated diffusion. All three are passive transport. All three involve the movement of a substance from an area of higher concentration to an area of lower concentration. So we talk about diffusion first. An area of higher concentration to an area of lower concentration is called a concentration gradient. Okay? So molecules are in motion, and that's the driving force of diffusion. So if you have a large concentration of molecules and they start moving randomly, you expect them to spread out. In other words, they're going to move from an area of higher concentration to an area of lower concentration. Let's say I bring in a can of tuna fish that's been sitting in my garage for a year, and it's swollen, and it's ripe, and it's nasty. Well, I bring a group in, and I turn off all the vents, and everybody's sitting there, and I pop the seal. Well, these molecules are going to diffuse rapidly in the air. I'm going to smell it first. Then pretty much everybody in the room is going to smell it. It's diffusing from an area of higher concentration to an area of lower concentration. Let's say somebody in the back of the room does the same thing with a can of potpourri. Well, those molecules are going to diffuse through the air as well along their concentration gradient, and then the whole room is going to smell a bit strange. So if things can diffuse through cell membranes, like oxygen and carbon dioxide, they will according to their concentration gradient. That's diffusion. Osmosis is diffusion also, but it's the diffusion of water molecules from an area of higher concentration of water to an area of lower concentration of water. Now, how do you get different concentrations of water? Simple, by having different concentrations of solutes in the water. So if you look carefully on pages 132 and 133, and I may even put this on the board here, let's say, hopefully you can see this on the video. Let's say we've got a tank of water, And we've got solution A and solution B, separated by a selectively permeable membrane. Let's say that solution A is 1% salt. Let's say solution B is 3% salt. And let's say that the salt can't pass through the membrane, but the water can. Now, if this is 1% salt, it's 99% water. And if this is 3% salt, it's 97% water. Now, we have a concentration gradient of salt, but it doesn't matter. Salt can't pass the membrane, but water can. So water is going to diffuse from the area of higher concentration of water to the area of lower concentration of water. In other words, the water is going to move toward the saltier side. Water follows salt. Now, terms in your notes include hypertonic, hypotonic, and isotonic. A hypertonic solution is a solution that has more dissolved solutes in it than another. Hypotonic means it has less dissolved solutes. It's more dilute. Notice the direction of osmosis. The movement of water across a selectively permeable membrane is the definition of osmosis. And it will always occur from the hypotonic side of the membrane to the hypertonic side of the membrane, whatever side that is. Okay? Now, of course, if cell membranes are subjected to this, they have to deal with it accordingly. Now, suppose both solutions had the same amount of dissolved solutes, in which case the same amount of concentration of water would also be the same. They would therefore be isotonic. With isotonic solutions, you do not expect osmosis to occur because there's no concentration gradient of water. They're equal. On the bottom of page 133, you'll need to pay careful attention to that. What happens to cells when they're subjected to hypertonic, isotonic, and hypotonic solutions? Now, keep in mind that red blood cells could take on water in a hypotonic solution and burst. But that won't happen to a plant cell because it's got a cell wall. It will build up pressure, known as turgor pressure. That's what happens when plants absorb water after they're wilting. Okay? Now, in a hypertonic environment, the cells will lose water and they will shrivel up. It's basically the same thing as somebody puts salt on a slug. Okay? That's osmosis is what's happening to that poor animal. So cells have to avoid these situations, and that's part of what water balance is all about in an organism. Facilitated diffusion is also passive transport, and this is the diffusion of substances across membranes with the help of transport proteins. In other words, the transport proteins allow for the diffusion of an otherwise impermeable substance, say sugar or whatever, if it's, I'm giving the accurate situation here. So it's still diffusion, it's still passive transport, but it's the use of membrane proteins to allow the diffusion of something that otherwise couldn't go through. Go back to the chain link fence. Chain link fence may have a gate. If it's open, you can roll a basketball through it. Facilitated diffusion. Active transport is the use of membrane proteins to pump or force molecules against their concentration gradient. So you're moving molecules from an area of lower concentration to an area of higher concentration. You're going against the flow. It takes energy to run a pump. So the sodium-potassium pump, which is found in all cell membranes, actively pumps out sodium and actively pumps in potassium on a continuous basis. That is active transport. So that requires energy. Bulk transport is when large amounts of things are brought in, or large things are brought in, or large things are moved out. Now, exocytosis is bulk transport out. Endocytosis is bulk transport in. Page 139 shows you bulk transport. Now, endocytosis can be of two kinds depending upon what is being brought in. Phagocytosis, Phagocytosis, is when a cell brings in food material or solid material. The word literally means cell eating, from the prefix phage, which means to eat. Pinocytosis, P-I-N-O, cytosis, means cell drinking. So if the cell's bringing in fluid instead of food, that's called penocytosis. So phagocytosis and penocytosis are both endocytosis. And that concludes the chapter on cell membrane structure and function.